Good afternoon, everyone. Um, as I was introduced, I'm from Maldives and I'm presenting part of my research. And this part is honey quality, to check honey quality. So, to check honey quality, the best reliable of one method is using physiochemical and microbiological evaluation. So, as we all know, every day we use honey in our daily life globally. And we know that it's a natural sweetener and it is produced from bee from nectar by collecting various plants nectars. So all the honey consumed in the Maldives is imported because in the Maldives all the food products are imported actually. We don't produce any. That includes honey as well. However, there's no study so far conducted about checking the high quality of honey. So we thought this is a very important research to conduct, even though we know that many studies have been done on quality of honey, but imported quality not done yet in the Maldives. So the aim of this study is to verify the quality of honey imported in the Maldives from various countries. The objective to achieve our aim, we have two objectives. That is, first to physiochemical characterize the honey, and then microbiological identification of this honey. Actually, since in Maldives we don't have laboratory facilities, we were forced to send the samples abroad. So we collect sample from the capital city, that is Male, and totally 34 samples we collected. That means including the duplicates. We collected from the main supermarkets. So this is just to show you the countries. So from totally about 10 countries, Australia, India, and this number is, that means 4 samples. Each brand we took 2 samples. The reason why we did not take more samples is because cost-wise, it's very expensive. So the samples first we collected from the market and then code and labeled the samples, and then we send it to the accredited laboratory. I think everyone knows that SGS laboratories. So we make sure the laboratory we are sent is accredited and their methods are standardized. So we had agreement with them, so send our samples, and they assessed these samples for us. So according to their procedures, it's ISO method or Sri Lankan standards method they have used. So these results we analyze using ANOVA just for the mean and standard deviations we checked. There's not much complicated analysis in this one. So I'll share the results. First, physiochemical characteristics of honey. This is just to show you overall all the physiochemical characters. So the one with the highest, that's 80% carbohydrate, and then total sugar is in brown one. So it's basically showing the countries and the brands and the carbohydrate, protein, age, moisture, total sugar content. I'll be giving each separately, so it will be a bit more clear for you. So the carbohydrate, we found its average is 80% from all the 34 samples, and it ranged from 79 to 82. It is similar. We found that Hakim from Indian researcher, he also had the same range of carbohydrates, and this is expected. This is just to show you all the brands. So as you can see, the below all the samples has more than 79% of carbohydrates. So we move to sugar. Sugar is very main characteristics or composition of honey, as we know. So it composes fructose and glucose, and the proportion of fructose to glucose uh, in honey relies on the nectar sources. So the quality also depends on fructose to glucose ratio. And we found that these samples contained about 1.1%, and the similar results was obtained from Nigerian honey as well. That's 1% range. And the fructose to glucose ratio is helpful. Why? Because it indicates the ability of honey to crystallize. So when fructose ratio is greater than glucose, the honey is fluid, as we all know, and the crystallization normally is slower if the honey ratio suffers from 1.3, and faster when the ratio is less. So the reducing sugar is another invert sugar, or primarily the fructose and glucose, and it's the main component, as I stated before. The average total reducing sugar found in our sample was 68%, and this is a bit slightly higher than the codex limit, that is 65%. And other researchers found that average reducing sugar, they checked only 5 samples, that is from Portugal, that is 71%. So that is higher than our value. And Indian researchers, they found their range as 71 to 80%. These are the indicators of maybe it's adulterated by sugar. Sugar has been added, artificial sugars. So the prevalent sugar in the tested sample for fructose had 35%, followed by glucose and maltose. And we found a few samples contain maltose higher. That is also suspicion that this honey has been, sugar has been added, which is not supposed to be added. So sucrose was found in one sample, but it is within the limit, that is less than 10%. The overall total sugar content is 70%. So the results are comparable to the honey from Benin and also Algeria. Uh, This is just to show you again how the flow is. So all the samples contained more than 50% sugar, total sugar. The next is protein, which is very small amount. Honey comprises about 0.5% protein and mainly enzyme and free amino acids. So total protein in the ones we checked is between 0.1 to 1.5%, and the mean range is 0.3%. And this is again just showing you the Sri Lankan product had 1.2 to 1.4%. The rest is within the range. So according to the codex, it has to be less than that. So ash content, when we move to, indicates the overall mineral content of honey, which is very low. The ash content of the honey samples in this study range about 0.0 to 1.4%, and the average is 0.1%. And the similar was observed by other researchers. And two samples, however, had 1.3% and 1.4%, which is above the codex limit again. That is, codex limit is it has to be no more than 0.6%. This is again, you can see it's the Lake Naturals, that brand is from Sri Lanka. So the moisture content we also checked, and we found it's of ranges from 14 to 20%, and the mean value is 18%. When we check the codex, it says moisture content should be not more than 20%. Moisture content of 17-25% honey was found in from Pakistan's honey. So this also shows how, highlighting to us that honey is available globally is adulterated or lower quality. So not all of them are high qualities. It is moisture content is vital as we know is it is stable against factors like fermentation and crystallizations, and a low moisture content help in protection against microbiological activities and preserves the product as well. So this is just showing the overall ones. As you can see, its moisture level is it's obvious. Most is above 15%. So hydroxymethyl furfural HMF, we all know that it's a freshness and purity indicator. We can use it. So we tested HMF in the honey samples and we found it range from 3.8 to 1000. So obviously there is exceed in range. The codex limit gives some samples it has to be less, it should not exceed 40 and definitely not 80. So we did find some of our samples. Only 8 samples we found it is within the range codex has permitted. And the samples that are above 500 mg we found it possibly it's adulterated with invert syrups or other sugars. So high HMF content indicates again poor storage condition or extensive heating of honey during its processing steps. So now I have completed the physiochemical properties. From there, we understand that some of the honey products are adulterated, and possibly by sugar. And the microbial analysis, we checked fecal coliform, salmonella, and we found that it's absent, which is a good news. These imported products are well preserved against microbiological contamination, and that the beekeepers did maintain hygienic practices during handling them. So the absence of fecal coliform has also been reported in honeys from. So we have seen that quality honeys is available again. So two kinds of honey is available in the market globally so far. So the absence of salmonella has been reported by others as well. Yeast and mold count in this study range from 4 times 10 to the power to 2.6, 10 to the power 5. Uh, and classic honey, two honeys had a bit higher ones, indicating that some kind of poor handling procedures is there maybe during the processing of honey. So similar results are also found in by other researchers, indicating again some of the products in the market that is available is not in highest quality. Presumptive bacillus series, we tested and found some are positive. It is associated naturally. This bacillus series might be present in the honey because it's the microflora of honey bee gut. So in the gut, this microorganism already is present. The result is comparable with the other studies as well. The authors explain that can be found regularly in honey, as I said before. It is not associated with food poisoning because it's not hazardous at very low amounts. But if it's high, then there is a problem. That's why we could not include the results. So the presence of bacillus may be due to its stability to survive in harsh condition. As we know, bacillus species are resistant spore formers. So even though honey is very stable, but it is normal to find this bacteria if it's not properly handled. Other researchers have isolated different species of it anyway. So in microbiological part, no microbial growth was detected in honeys. Especially we found that none of them detected in Malaysian, Australia, Spain, and Indonesian brands. This is just to coliform not detected. as some of the fungal identification is there, like aspergillus found in some of the brands. And this is just giving some ideas about what we detected from that one. Some bacillus species, fungal and eastern mold. So the conclusion is we tested 34 imported honey samples from the molded supermarkets, and we checked the quality based on physiochemical and microbiological parameters. And the honey imported molded is generally, we can conclude generally it's in good quality, even though there are some issues such as some of the products have high HMF, and the honey samples were contaminated. Some of them are contaminated with microbes such as Bacillus, yeast, and Aspergillus species. And the presence of this bacteria is it is indicating that better, proper, and cleaning and handling is required. And during processing steps, more hygienic steps needs to be required. So the results suggest that there may be some values in conduct in random sampling. That is, in the mold is entry points we need to control. So during the authorities, for example, health ministry, when they do the random check-in, they need to take samples randomly and send for sampling to check the quality. So this is what we found, that there is a need for this check. This study is funded by Maldives National University grant. So I would like to acknowledge. Thank you. Any questions?